Hi, and you're very welcome to our latest Think Limitless conversation. And we're joined today by Professor Lokesh Joshi from University of Galway. Hi, Lokesh.
Hi, John.
So at Carelon, we build great software and AI solutions for better healthcare. And this LinkedIn Live conversation is available live on LinkedIn this afternoon, but you can also watch it on replay. If you are joining us live, you're able to ask questions on the LinkedIn chat. So if you have a question along the way during the conversation, please take the opportunity to ask, and we'll endeavor to answer. So it's a 30-minute conversation, and it's wonderful that you've joined us today, Lokesh. We've known each other for several years, and it's amazing the work that you're doing. Well, what I'd like to do is just maybe give an introduction to you for the benefit of our audience, an introduction of your background. So you're currently Professor of Glycosciences at National University of Ireland in Galway. And you go back to your education days, you have both a degree and a master's degree in life sciences from the University of Rajasthan. And you have a PhD in microbial pathology from Bath University. You're 16 years in your current role, or a progression of roles rather, with Galway University. But prior to that, you were 10 years with Arizona State University, where you were a professor, and prior 6 years at Cornell, where you were a postdoctoral fellow. You have an illustrious career and extraordinary area of research currently underway, and we're so delighted you've joined us today, Lokesh, on our conversation. I suppose to start our conversation, one of the things that we've talked about quite a bit, you and I, is around the research you're doing. So I guess, for the benefit of the audience, maybe tell us a little bit about the research that you're currently undertaking.
Thank you, John. I really appreciate the opportunity to speak with you today. I think it's really incredible to have these, opportunities when we can talk, where people come from different disciplines. I speak to people in biological sciences, life sciences almost every day, but it's great to talk to you about some of the things we are doing and maybe learn from you, and maybe think of making progress not just for my own lab but also for the field in general. So I think this is great for me as well. So in terms of my research, really, over 30, 35 years, my real interest is that how do cells talk to each other? So when we think of our cells or any other cells, we just see them, as they are, but there is a constant communication going on. Every cell is a sensor, is sensing something, and then it responds to that. So there's communication. So we became, a few billion years ago, from single cellular to multicellular organisms. Then there was more need to communicate, to build the tissues, organs, organisms, also to identify us versus non-self, and to talk to each other. So I think that's something that I've been very interested in, and then that led me to what is the kind of the area of fun, which is the intersection between two cells, what we call the cell surface. And that's where really the interesting things happen, because you have molecules there, and they are used as the chemical codes on every cell, and they talk to each other all the time. Like you and I are talking in English right now, but if you were to switch to Irish or French, I'll lose most of the communication. I'll lose it, right? So that's what happens. If the cells lose these molecules, or the string of these molecules, or the structure of the molecules, then miscommunication happens. And that's how diseases happen, is the miscommunication between cells, whether it could be our own cells, it could be a good bacteria, a bad bacteria, whichever way. And that's how most diseases start. So I'm very interested in how does the communication take place. And that has really led myself and my team and people in my field into various different areas, such as biomarker discovery, understanding cancer, inflammation, infection, all those wonderful things.
And when you think about the impact, then, as you're alluding to there, the impact on people, the potential benefits from your research. You've talked a lot about that as well, in our previous conversations. But maybe tell us a little bit more about that. What's the impact to real people of the research?
Yeah, and I think that's one thing. Again, we need to I guess this is just my personal feeling, John. I think all of us need to really take it very seriously that it's up to us to find solutions as well. There are a lot of challenges. We can focus on the challenges, but there are solutions as well. So as a scientist, I've always thought, okay, so what could I bring to the humankind? Yes, one can write more papers, more books, more all those things, but what are we bringing to people in general? So where my group, my team, and also a small startup company we've focused is really that nature is the largest and the oldest lab on this planet. 3.8 billion years of evolution has been taking place. Could we learn from nature to protect ourselves from the infectious diseases, from pathogenic organisms, whether they are viruses, bacteria, fungi? How do we protect ourselves? So we've gone back to nature. We've seen how does nature do it every day, in and out, as I'm breathing, as you're drinking, eating, we're inhaling and taking pathogens, but we are not getting sick every day. So we want to learn from that. And that's what we've done over the last 10, 15 years and bring solutions to people that they can use to protect themselves against any pathogens, whether it's a virus or a bacteria or a fungus.
Fantastic. And you've often talked about the parallel between computer language, the coding at the heart of computer coding itself, ones and zeros ultimately at its most basic level. The parallel between that and cellular language, biochemical language, but proteins and sugars. Maybe tell us a bit more about that, how you see that parallel.
Yeah, and again, look, I'll be the first to admit that I don't understand computer science, information technology, any of those things, but I have this thought that I see, especially the sugars and proteins on the cell surface, because that's my field, that they're strung together for a reason, and they're strung together to give a message to other cells, right? So that's the code that the cell is presenting to the environment. So, and if that code changes, if that sequence ones and zeros of sugars and proteins changes, it becomes a completely different structure and different message. So similarly in computer science, you would have zeros and ones. And if something goes, if there's a mutation, if one of the zeros is missing or one is missing, then the whole sequence changes. Similarly, in biochemistry, molecular biology, if one of the pieces of DNA, RNA, protein, sugar is missing, it affects the whole thing. And that leads to, as I said, miscommunication and wrong messages and mutations. So I think there are a lot of parallels that we can learn. And even the terminology such as self versus non-self, a computer is there, a virus enters. Now, in biology also, there are many viruses and bacteria that actually pretend to be ourselves. They take on the same codes, same sugars, and same proteins as we have, and they go under the radar and cause damage. So, how do we identify those? So, I do think that there are a lot of parallels in computer science, information science, and biological mechanisms in the body.
Right. And when you think of current infectious diseases, you talked about it there. Ultimately, the benefit of your research, as you've described it, is predict and prevent illness, and preparing people in a more, I suppose at one level it's a mindset change, but also it's therapeutic as well, isn't it?
Yeah, no, 100% right, John. I think when we talk about predict and prevent, I think right now, at least in my field, we're very much experiment driven. But I think, and yes, we'll find the solutions. We have found the solution, but it's going to take us a long time. But I think if we could collaborate with experts in machine learning, artificial intelligence, large language models, I think we accelerate that pace of discovery and find solutions. And then they can be tested in the lab, really before they're deployed, to whatever disease it is. So I think a collaboration could accelerate biomarker discovery of novel drug targets, but also novel drugs. But in my field, what we're looking at is how do pathogens attach to cells? How do they cause infections? If we understand that better, then we can develop better molecules, or we can find better molecules in the nature already and use them to protect people and animals.
And part of that, I guess, is back to the area of artificial intelligence, machine learning in particular, other data analytics tools to model and understand cell to cell communication. And that's an area of emerging research too, isn't it? That how can we bring machine learning capabilities to bear on the question that you've raised?
Yeah, again, look, I think in some ways it's very obvious. So scientists are working anything from US to Australia, all over the place, north to south, doing really good experiments, publishing a lot of papers, a lot of information is out there. But I think what we need to maybe look into is ways to collate that information, verify the information, because otherwise the labs work, a lot of times labs can work in silos, unaware of what's happening. So if we were to bring the information together, even if you were to focus on cell-to-cell communication, my belief is that a lot of data is already there. We just haven't gone looking for it. So in some ways, in my mind, like 100 years ago, people were looking for gold mines, but I think the data is the gold mine now,
Yeah.
right? How do we find those gold mines? How do we trawl through the data? How do we find meaning in that? And how do we use that data? That is the challenge now.
And a number of years ago, Lokesh, you gave a TEDx talk at TEDx Galway, and that had a huge following. It sparked a lot of conversation. You talked about glycoscience as the sweet and sticky language of life. Is there anything you want to tell us about that? Your memory of that event and the follow on from that TEDx event in Galway?
Yeah, no, then. So studying sugars and proteins has been my passion for a long time. And I think what I was trying to convey in that TEDx talk is that, especially the study of sugars, when we think of sugars, we think of sweet things, we think of diabetes, but there are many different kinds of sugars that are not sweet, they're absolutely bitter. And then in fact, there are more sugars on the planet than anything else on this planet. And nature doesn't waste things. There's a reason why nature produces something. So if you think of even cellulose or chitin, which is found in shellfish and insects, they are the most abundant biomaterials on this planet. And so it can have many functions. So the idea there was that if you look at a bacterial cell to our cells, they're all coated in sugars of different kinds. And they're very complex structures, but they play very important roles. There's a slide, and this is a different slide talking about, this is what I call self versus non-self. That you take, researchers took two sponges. There's one is red, one is white, and they macerated them, they ground them up, put them in a beaker, and the red went to red and white went to white because they identify each other based on the sugars and proteins on the surfaces. They become the nanoscale Velcro that are specific to specific cells or species. So to me, it's a fascinating field, but we haven't really gone into the information side of that yet. We are looking at the biochemical and molecular aspects only. And here's another slide on my left here, which is the model of the cell surface. The red bits are the sugars, and the kind of floaty light blue bits are the proteins. So they coat the surface like, we have hair all over our body. So that's what every cell has, this, and every one of them is a receptor, is almost a kind of, it transduces signals. So on my right hand side here, there are two cells. It's a 2D model, and how these proteins and sugars are used for cells to talk to each other, which is the top and the bottom of the right side, but also for a cell to talk to virus, bacteria, which a parasite like amoeba, toxins. They all attach to us using sugars and proteins. So if we can understand what they're attaching to, then we can develop modalities or solutions to prevent the attachment as well.
Wow. And you also talk about, when we get into the genetics of this, that, the total number of genes does not define complexity. So there are forms of life on Earth which have more genes than humans, for example.
Yeah. So if you were to look at it, John, that's almost funny thing, because when we talk about total number of genes, we have approximately 20,000 genes, in fact, less than that now. But also we have the whole kind of dark genome as well. This is only expressed genome. So there's a whole different context there. But even in terms of number of expressed genes, we have less genes than some plants do. We have less genes than onions, So that doesn't mean that we're less smarter than onions. I'm sure onion is smart in its own way. Like every species is smart and fits in the nature for its own place. But number of genes doesn't define or doesn't equate to the intelligence of the organism.
But, when you think of genetic variety in the human race, it's very narrow. So all people in the whole world differ by I think is it 0.1% genetically. It's something very, very narrow,
That's correct. Yeah.
indicative of a bottleneck event, maybe estimated over 70,000 years ago. I think isn't that a theory
That's
of
right.
how that occurred? So we are truly all brothers and sisters on this planet.
Absolutely right. Yeah. And I think what makes a big difference is what they call now epigenetic differences, the differences that are on top of the genes and that are not written in the blueprint of our DNA. That's what happens once the DNA is transcribed into proteins. So I think this is where the nature versus nurture also comes in. But you are absolutely right that end of the day, we're all brothers and sisters as a species on this planet.
Yeah, and we have a question from Liam. Um, does this overlap with organ transplant rejection drugs? I suppose, yeah. Does the area of your research
Yeah, yeah,
overlap with that
yeah.
area of?
That's and sorry to interject. I think that's 100% right. So there is a sugar molecule called alpha gal, which we don't have, but other animals, other known primates have it, and some bacterial viruses have it. In fact, some of the parasites have it, and the bacteria that causes Lyme disease has it. So pigs have that particular sugar that we don't have. So when especially pig organs are transplanted into humans, it causes acute rejection because our immune system thinks it's a foreign body. So even to the point that our blood groups A, B, O, and AB are really different in just one sugar, right? So you can just imagine that although it's only one, what we call hexose structure, but it is so potent it can lead to an immune reaction or rejection.
Yes, and when you think of, I suppose, with that as well, when back to the very narrow genetic difference between all humans on the planet, it, part of the danger of that very narrow variety in the human race is that certain diseases can spread like wildfire because the genetic variety is too narrow in the human race. COVID, perhaps an example, is it, of that, where everyone is susceptible. Um, there's nobody innately immune to COVID.
Yeah, no, 100% right. I think, again, unfortunately, it's not the last pandemic we'll see in this century.
Yeah.
I think that's something, again, I go back to the fact that, okay, biologists and computer scientists and other types of scientists need to come together to find solutions, not for today's problems, but also future potential diseases that we might be encountering.
I mean, you're talking about as well climate change and the impact of a world which is becoming very different. That the for maybe 12,000 years or so, the Earth's climate system, ocean currents, air currents, etc., has largely been operating in a stable way, but that can radically change if the climate changes, and then infections can spread from geography to geography, where previously that was harder. The melting permafrost being one example that people are nervous about. Deforestation, is that something that we need to be more anxious about, do you think, Lokesh?
Yeah, John, I know we shouldn't use the word anxious, but it's kind of an innate response that we all have. I think we should be aware of those things. We should also be aware of almost a non-linear relationship of events and what might happen as a result of those events. So if you think of, climate change, or carbon dioxide or higher temperatures, they have major impact, not just on non-physical bodies such as the mountains or rivers or lakes, but also on the biological beings, because these non-biological things are composed of biological elements. So if the glacial ice is melting, they are reports, and again, one just has to be aware of them, that they are harboring millions of years old or thousands of years old microbes in there. Now, 90% of that might just be the fodder for the sea, but even a small fraction of that, if that's alive and if that happens to be pathogenic, that's something that we have never experienced before, right?
Yeah.
Similarly, even if you do look at the ones that are existing right now, not the new ones, you can see that the geographical range of pathogens and pests is shifting because temperatures are rising in the places where there wasn't that warm temperatures. So, in fact, I remember reading late last year that they found Zika in France. Now, it's one of those things. The whole world is shifting now. You see mosquito, you see malaria, you see, also different diseases. So we are living in a world where climate change is going to affect the disease spectrum that we'll be encountering. And some of those we may not have seen before, or we may not be prepared for them.
Yeah. We have another question from Liam, same Liam. For you, Lokesh, are you using technology that generates proteins and sugars combinations, I guess, to simulate a cell coated with them?
Yeah, it's an interesting question, Liam. So I don't want to go into the details of the science here, but there are people in my field who are looking at some of those things, where you can feed cells different, precursor molecules, and they generate certain molecules on the surface. But what we are looking at is looking at natural decoys that will then bind to the pathogens and remove them from any surface or any cells. And
Yeah.
what we want to do, and again, I feel this, I could be wrong, I could be naive here, that any company should really be first and foremost an environmental company. And then we can become a company of medical devices, medical technologies, diagnostic, because we want to make sure that we don't harm the environment. So one thing we are trying to do is to develop technologies that are completely biodegradable and safe, not just for us, but all organisms, whether it's aquatic or soil or the environment. But that's how we need to think in 21st century, is how do we come up with, completely benign solutions that don't harm anybody around us.
There's also another emerging area of understanding around the connection between viruses and certain cancers. Certain viruses seem to trigger cancer. The Pamplova virus and cervical cancer, as an example.
Yeah, no, I think you're absolutely right. Now, it's an interesting thing. So when we look at each other, John, we think of two individuals, but we're really ecosystems. There's a John ecosystem, there's a Lokesh ecosystem there, right? So we may have, let's say, 3 trillion cells of ourselves, but we have a 3.5 or whatever number of trillions of microbes in and around us. Now, 99%, or maybe more than that, they are completely benign. In fact, they're helpful for our body's functions, and they help us in all different ways from the time we evolved as animals. But there's a small percentage of them that can be harmful, that can be deceptive, that can cause damage, in different parts of the body. So it is being proven now that there are certain viruses that are affecting the cells to mutate faster, or they insert their genes into our cells that mutate, and that leads to cancers. And
Mm,
in fact, there is hypothesis that there are a lot more cancers that are caused by viral infections than just a cervical cancer.
right. And
Yeah.
I guess modeling that, I mean, one of the challenges has been that with conventional computing, it's quite difficult to model interactions. You can define a model, but conventional computing struggles to deliver in a timely way a predicted outcome. And this is something. I mean, it's not you know it's not an area of your research. I don't understand. I don't. I believe currently, I I, I think it's not an area, but the area of quantum computing,
Yes.
which people are now beginning to talk about in the realm of vaccine discovery. That
Yeah.
quantum computing, by its nature, comes at the modeling process in a different way, which may yield timely results in a way conventional computing can't.
No, 100% right. Actually, I think we are at the very early stages of kind of high throughput, accelerated discovery aided by computing. I
Yeah,
think what we're doing so right now, and you are the expert there, I think maybe not fast enough, but if we bring quantum computing, we can do things in the matter of minutes or seconds that will take us days or years, right?
yeah,
No, I actually really think that we are at the intersection right now where if computing and biology came together, if the scientists came together, if they were able to communicate properly, because sometimes there's a communication barrier, right, between the two types of scientists.
mm,
If we were able to communicate better, then I think we can find solutions much faster. And as I said, then things go back in the laboratory where they are tested, whether our discoveries are right or not, and then we can then look at solutions. Yeah, I would absolutely believe that modeling is possible. So, for example, what DeepMind has done with protein folding,
yeah,
that's just the beginning of demonstration that how computing and AI can help us understanding biology. But then you can go to the next level of, protein-protein interactions, protein-sugar interactions, and all those different things, start to build models. But again, in my case, what we are trying to look at is that what's available in nature, how can we bring them in, how can we model them, how can we study them, and how can we develop something that can help people right now. So that's something that we've been able to do.
There's a sense that the race is on. There's this race between finding solutions using our, as you say, alignment of different scientific branches, computer science, life science,
Yes.
on the one hand, and then the other hand, this accelerating evolution of viruses and other pathogens for all kinds of reasons. So the race is on. There's a sense there's a race that's on between the two forces.
Yeah, and again, that's an interesting thing. You mentioned that because in evolutionary biology, there's a term called red queen effect, where the red queen is running but going nowhere. So I think that's what happens in evolution as well, is that we are always running, and so are the other pathogens. Just trying, this is a race, so who wins and who loses? So they are adapting, as you mentioned the word adaptation before. They're adapting all the time to host, but then we have to adapt in a way to continue to identify them and control them or remove them from our body. So the adaptation and genetic changes and changes in sugars and proteins happening all the time. And I say all the time, it doesn't happen in one generation, it happens in multiple generations. But that is the evolutionary process, really. Is a race, basically. And again, sometimes, so what's happening even in the different variants of COVID is that it is mutating all the time, to the point that, we think, okay, so what will be the next variant? Will our vaccines work against it or not? How is it infecting us? Will it be more transmissible? Will it be more aggressive? That's all because the race of mutation there. But we can't mutate fast enough, but they can mutate very fast.
We're down to our last 4 minutes, and it's amazing how the time flies in this engaging conversation. And I wanted to ask you a little bit about your company, Aquila. So in addition to being professor at University of Galway, you also founded a number of years ago Aquila. Maybe tell us a little bit about that, Lokesh.
Yeah, again, so I'm driven by bringing something to the humankind. And I just feel that there's a responsibility on all of us, especially those who've been privileged to have higher education, research background, and all those things, is what can we do with our science and bring it to people? So Aquila Bioscience actually started working with the Defence Forces Ireland to begin with. Then we worked with the European Defence Agency, the Czech Defence Forces, to develop technologies to protect the soldiers against certain biological threats. But the implications of the technology are for everybody, from a little baby to an elderly person who may have infections, who may have some skin conditions. So what we are looking at now is the impact of Aquila's technology, again, based on how nature protects us. It's called biomimicry, or nature-inspired technology, and how we can bring that in the lab and make something and bring it to people. They can use it, day in and day out, without having to rely on chemicals. Because in my perspective, we've been on this chemical train for 100 years, and we have, in fact, only yes, we have saved lives, but we've created a lot of antimicrobial resistant bacteria, multi-drug resistant fungi, and all those things. We can't continue to rely on synthetic chemicals. We need to think outside the box, like your talk of thinking limitless. Our kind of thinking is that thinking outside the box. Let's see how nature is doing it, and then let's see how we can find some solutions for people in a way that's sustainable and effective.
Wow, that's fantastic, Lokesh. Very inspiring mission and sense of purpose with your company to go after that. It's incredible. We're down to our last couple of minutes. Is there anything else, Lokesh, you want to tell us about what's coming up? As we look across the year ahead, 2024, is there anything that you want to tell us about that's on its way that might be of interest?
No, John, I think the only hope I have is that I think we can create more collaborative ties, because I think innovation happens at the intersection of different expertise. So as I mentioned, the cell-cell communication is the interface of two cells. That's where they talk, that's where they shake hands. And that's where the magic happens. Likewise, if people who came from different backgrounds and talk to each other, that's where the magic happens. If you live within yourself and within your own community, within your own discipline, that's where things don't really improve. So it's the outliers that bring the change. And I think we need to really basically step out of our own comfort zone or zone of comfort and just bring the innovation out to people.
Yeah, it's a fabulous thought to end on. I mean, we're right up at the half hour, Lokesh. We'd like to get you back on future Think Limitless conversations. We'll definitely keep in touch. And thank you again for taking the time today to speak with us, Lokesh. We really appreciate it.
Thank you, John.
Thank
I
you.
appreciate the opportunity. Thank you very much.
Thank you. And thank you, everybody, for joining us today. And tune in again. Next Think Limitless conversation will be in about 2 weeks' time. Take care, everybody. Thank you. Bye now.
Thank you.